Good morning, Mr. Kirasal.
Hey, good morning, Matthew. Good morning, folks. Welcome in. Hope you're having a good day so far. Good morning, Jessica. Good morning, Dean. Good morning, Colin, Stanley, Sarah, Rose. What's happening, folks? What's happening? I gotta get my socks on. Look at how cool my socks are. They're so funky. Handmade. These are my cozy socks. Even though it's like boiling hot in my room, my feet are cold. What else is new? Thanks, Matthew. Alrighty. We got a bit more of a classical day today where I will be teaching to you. We are learning some new concepts today on top of some building concepts. So we got a fun, happy Friday indeed. We have a Friday planned and, we got a lot to do today, so to speak. So we'll make sure we're maximizing our time as we get the day started. I need to find, where is my meiosis review? 1 second. Morning, Rocco. Good morning, Ava. Even though I said happy Friday, that was a response to you. I also meant to say good morning. Okay, where is my meiosis chart? Why am I struggling to find that? Let me see if I can find it. Meiosis review. This is not it, I suspect. Morning, Maha. Once again, good morning, everybody. We'll get started in about 3 or 4 minutes. We're going to get everyone in before we get started just because we have a jam-packed day today. And yeah, we just got a busy day today. You're going to be needing a piece of paper. You're going to be needing a piece of paper. You're going to do a little bit of drawing today. Not so much drawing in the classical sense, but you're going to have to create your own chart. So a nice big blank piece of paper if you got it. Line paper will work as well. But I'll reiterate that at the start once we get started because I forgot as I'm looking at my notes. I was supposed to give you a chart, but in my notes I said I want you to draw it instead of copying it in. So it'll work out nicely. Or do I want to give you all a chart? I'm still deciding. What is this one? Yeah, I'm definitely going to make you draw it, I think. No worries. Alrighty. Where's my Zoom? There it is. Alrighty. So we'll wait 1 more minute, get some people in. We'll take a look at our schedule for today. Oops. Here we go. Here we go. Here we go. Alrighty, let me get the schedule up. We'll take a look at what our plan is for the day and we can get started. So yesterday we looked at mitosis in a little bit more detail. We spent some time on that. Application on Explore Learning to kind of get a feel for it. Well, that was 2 days ago, sorry. Yesterday was our house day / feedback day. Today we are looking at sexual reproduction. And in order to look at sexual reproduction, we have to study the process that is called meiosis. Now, this is classically a little bit more of a brand new concept in the context of it could be a little bit tricky, but if you get through the lesson, you write it down, write good notes, make good notes, type them out, whatever, draw good diagrams. As I said right off the top, you're going to need a sheet of blank paper. I'll put that here just to remind. sheet of blank paper needed. Just because I want you to draw in and write down some notes. Write down, not type out. I want people to draw these diagrams by hand. I don't care if they're ugly, beautiful, perfect, imperfect. I just really want you to get that idea connected with what you're drawing in order to get this into our brains, so to speak. So if you don't have a sheet of paper yet, go get one. I'll wait a minute until you got it, and then we can get started on our lesson today, because we are learning a brand new concept. We're learning a brand new concept in the context of sexual reproduction. So get that blank sheet of paper if you haven't gotten it already. You're going to be doing a little bit of drawing for me towards the end of the class. And again, it's okay. It does not need to be good. Everyone's always like, oh, but sir, I suck at drawing. Yeah, me too. Me too. It's okay. The important thing is that you give it a shot and you try drawing it out. Because if you don't try, then, well, it might just make things a little bit easier for you in the now. But this is the whole premise of that, is to make it a little bit more challenging now when it's easy to make it challenging. And then as we go on, you can make it a little bit easier for yourself. So again, those of you who just joined, good morning. We're going to be needing a sheet of blank paper. Line paper will work fine, but if you have a piece of blank paper, like a blank piece of computer printer paper or whatever you got kicking around, a notebook, anything will work, but you're going to be doing a little drawing and writing. Okay, let me take a look at attendance. We'll do some attendance. Then we can get started. Amina's here, Anna's here, Ava's here, Carson's here, Colin, Nicole, Yella's here, Dean's here, Emma's here, Fayang, Jessica, no Josh, Katie, Krish, Maha, Mariam, Mateo's here, Matthew's here, Olivia's here, Ratika, Rocco's here, Rose is here, Sahil, Sarah, Stanley, who's after Stanley? Sophia, Talia, Valeria, and Vicky. Sweet. Alrighty, folks. Ready to roll? You got your blank sheet of paper, you got your pen ready. If you're typing out the first little bit, that's cool. But without further ado, let's get started and take a look at lesson 3, sexual reproduction and meiosis. So the big reason we look at sexual reproduction, outside of like the obvious that humans reproduce sexually, as well as many organisms that we've looked at reproducing sexually, but the key thing here with regards to sexual reproduction in terms of genetics is some of the ideas that we're going to discuss here. Because the way sexual reproduction works, and the beautiful thing about genetics, is that we can make that direct connection. For those of you with siblings, you can look at your siblings, myself included. I have 2 younger brothers. I can look at my brothers, you can look at your siblings, and you can see directly that you are related, but you are not perfect clones of each other. You are not identical to each other. And that's as a result of the processes that go on in sexual reproduction. Because in asexual reproduction, or in mitosis or binary fission, yeah, I'm better looking, says Matthew. Yeah, that's a good. But again, right? Those are the things you can see and you can compare. Marianne's here, excellent. That you can compare with your siblings and in general, just look at those comparisons. Because like I said, in asexual reproduction, binary fission, mitosis, what have you, we have the one parent producing 2 identical cells. There's no variation. It's the same DNA. And it's usually found in those unicellular organisms, or mitosis, which is a form of cellular reproduction for specific types of cells. So you'll see that it is an exact copy, a perfect one-to-one copy. Okay, perfect one-to-one copy. With meiosis, or with sexual reproduction, we have 2 parents that produce genetically different offspring almost every single time. Without fail, unless it's identical twins, and even that's a little bit of a dicey concept, but without fail, Sexual reproduction will always produce a genetically different offspring, and it will always involve some type of special sex cell for multicellular organisms. In humans and most animals, it is sperm and egg, and we'll look at some of those processes for the creation of sperm and egg throughout this course, as well as why it's so important and crucial in the context of sexual reproduction and genetics as a whole. Alrighty. So please recall, in the 2 days that we looked at mitosis over the last few days, actually, well, are people still writing this down? I'm not sure. I use the participants tab to kind of get feedback. Does anyone have any questions with regards to that? Still writing. Okay. When you are done writing, can you give me a check mark in that participants tab just to give me that understanding? If you're still writing, put a X there, because I know many of you are still writing and I don't really want to double talk. So yeah, keep going. Perfect. Okay, I'll let y'all keep writing. We'll give you a couple more minutes. And if you have questions, you can ask. Otherwise, keep going. Questions. Is the sexual reproduction, are the daughter cells identical? No. In sexual reproduction, we don't actually have daughter cells. And I'll talk about what that is in a bit. But there are no daughter cells in sexual reproduction. It's a completely different process and a completely different production. Correct. You're just taking the notes for now. And then I'll move on once everyone gives me the check mark, because some of you have given me the X, which makes me think that you're still going. I'm going to have some of my banana that I didn't finish for my breakfast. Potassium. All right, almost done. Another 1 minute. Bananas are delicious. Jessica asks, how can identical twins be different even if they are identical? Is it their DNA? I will talk about that in a little bit, Jessica. From birth, identical twins have the exact same DNA. And then as soon as they are exposed to the environment, processes that change your DNA take place. Everyone has it. It's not something we go into too much detail, but ultimately over the course of their life, there are subtle changes that are made to DNA as you move through your life. And twins, even though they're identical at birth or in vitro, once they are born, they are exposed to, for the most part, the same experiences. But as those things change and as they vary, they're exposed to external environmental factors, which actually, in fact, change their DNA on some level. And that causes them to eventually become different from their twin genetically. Is that why pretty much everyone born with blue eyes and then that changes later on? Yes and no. Yes and no. It's similar concept though, Ava. Alrighty. Almost everyone's given me the check mark thumbs up. we'll move on to the next part. And if you're just continuing along, that's okay. You can continue along as we go. So recall. Or how people's hair can be blonde when they're toddlers and then goes on to brown later on. It's similar. It's similar. Again, it's more to do with that's actually due to your genetics specifically, not necessarily to environmental factors, but environmental factors do have an impact on that as well. All right. So recall, mitosis is a form of asexual reproduction. It will produce two identical daughter cells from one single parent cell. Again, PMAT, prophase, metaphase, anaphase, telophase, that form of mitosis, cytokinesis, and interphase, all of those productions allowing for that asexual reproduction of those two identical daughter cells. These cells are diploid, and each somatic or body cell, aka non-egg or sperm cell, and that's a very important to distinct, right? These somatic cells, this is a brand new word for us. Somatic cells or body cells, right? Skin cells, liver cells, heart cells, brain cells, bone cells, muscle cells. Those are all somatic, okay? It contains two sets of chromosomes. In humans, the diploid number is 46, and we typically express this as 2n equals 46 for humans. So I'm going to just hang tight here for a second just to kind of get this point hammered home, so to speak. Oh, that's not the color I want. And then I want to talk a little bit about some of the arithmetic involved with regards to genetics, because we will incorporate a little bit of math as we move through this unit. Specifically just arithmetic and probabilistic aspects, nothing too big. So somatic cells is like the special one trick cell for our body. Not necessarily one trick cell. Oh, wait, I see what you mean. Yes. Yeah, it's the cell that just does one thing. But stem cells are also somatic cells. So not quite. Those are just, the way I like to call them is that somatic cells are your body cells. Whereas egg and sperm, those are the only two cells that are non-somatic. Okay. Yes, stem cells can turn into anything, but they are still somatic cells because they have diploid. They're two sets of chromosomal information. It's the diploid or haploid aspect that matters. Sorry, so good question though, Matthew. Okay, so two sets of 23. So I ask you, those of you who like your math, what does n equal? What does n equal? Beauty, 23. So if the diploid number, or 2n, if the diploid number, or 2n is 46, our haploid number, or n, is equal to 23. Does everyone understand that, what I mean by that? Yeah, n is equal to 23, or our haploid number is 23. Haploid means half, correct. Diploid means double, triploid, polyploid, multiple. If there was such a species that was quadploid, it would be 4n. Diploid, 2n, haploid, 1n, or half of an n, or half of diploid, or whatever you want to consider it. But haploid just means half in the context of having the number of total chromosomes that humans have. Okay, so we have haploid. Ooh, that's not what I wanted. Oh, well, that's fine. All right, any questions before I move on to the next part? Oops. Any questions before I move on to the next part? Okay, 1 second. I see some people asking stuff in the chat. Excellent. Excellent. Just give me 1 second. All right. No questions. How would you show polyploid? You would show polyploid based on the number of sets of chromosomes. So if I gave you a question like a polyploid strawberry is 10 times the normal size, meaning it has 10 sets of its DNA, you would say 10N for that polyploid specific. But the polyploid is, poly is just a multiple of, or more than 4, I should say. So if I say it has 6, then you would just say hexaploid. But we don't really get into that stuff, so you don't have to worry about it, Jessica. But good question all the same. All right, let's move on to the next part. Thank you once again for everyone telling me feedback-wise in the participants tab. Let's take a look at why this is an important concept to study. So the reason why we look at this is because this one set of chromosome aspect, actually, I want to wipe this out first. We'll let you get to that eventually, don't worry. So one set of chromosomes came from each of our biological parents' gamete cells. Gamete cells or egg and sperm cells. These are our sex cells. 1 second, we got a new joiner. Get them in there, bam. 20, what did I miss? Oh, we got it, okay, sweet. So this haploid number, or these gamete cells, also known as egg and sperm, are haploid. So gametes are called haploid because they only contain one set of chromosomes. As we learned above, we express this as N is equal to 23. For humans specifically, it's different for each species. And N is representing how many chromosomes are in a single set. So we get one set from mom, 23 from mom, and one set from dad, or 23 from dad. So we express this gamete haploid number as N equals 23. Okay, so haploid versus diploid, again, coming up. And now as we start to get into more detail about sexual reproduction, we can start to piece together some of the things we learned about plants and fungi in our last unit. And we'll talk a little bit about it in this lesson, or in this unit, sorry, as well as next unit. Is it possible for the mom to have one less and the dad to have one more? Yes, we'll get into the disjunction disorders, genetic disorders, a little bit later on. And we'll talk about why that's a bad thing, Colin. Or how it can be a bad thing, I should say. So haploid and diploid, haploid and diploid. So these gametes or sex cells, or egg and sperm in humans and animals, they come together to form what's called a zygote. So the zygote is a cell that forms when two gametes fuse together in the process that we know as fertilization. This zygote is the first cell of a new diploid individual. So when fertilization happens, the egg and the sperm come together. So if you recall from that note just above, the egg is haploid, the sperm is haploid. When they come together and fertilize, they can join their two genetic materials, one from mom, one from dad, together to form a fertilized zygote, which is now gone from haploid in the sperm and the egg here. It's gone from there, haploid, to diploid, where it will be the first formed cell in a new diploid individual that will develop into a fetus, which will develop into a baby, and which will be born into the world to be a human being just like you and I. So this process allows for that genetic diversity that we'll be kind of using as our main focus for this unit and next to occur, because it takes half of our DNA from mom, half of the DNA from dad, and it makes a brand new individual as a result. That zygote then goes through mitosis to form the trillions of cells that make up you. All righty, questions? So this is a brand new idea. Oh, I mean, maybe not a brand new idea. Some of you may have known this is how it works already, but the specifics of how it works, now we're getting into the nitty gritty, so to speak. Alrighty, questions? I'm still confused at what diploid is. That's okay. We can talk about it. Okay, so, oops. So we have two cells. We have two cells here. One has this genetic information, and one has this genetic information. Which one is haploid and which one is diploid? Just by looking at that chart or this diagram that I've drawn here for you. Which is haploid and which is diploid? Di means two. Left haploid, right diploid. So haploid and diploid. Ooh, I'll type that out actually. Now, how did you know that? Well, many of you just saw that this one on the right has two sets. The one on the left has one set. There's no midpoint, so I assume one is hap because it's hap. No sibling, yeah. It's happy on its own, one set. Okay, so haploid, diploid. Cool. Oh. All right, now let's try this one. Don't answer in the general chat. I see people typing. Oh my God, don't answer in the general chat. If you're doing it already, oh boy. Okay. So I've added an extra wrinkle in. Which one is haploid? Which one is diploid? Message me privately in the chat. Don't message it in the general chat. I want to see if people can get it. Which one is haploid? Which one is diploid? Okay, most of us seem to be getting it. Okay, so haploid means half. Haploid means half. Some of you are guessing it. Good, keep going. Okay, which one on the left? Which one on the left? What is it? Haploid or diploid? Good, good. The one on the left is haploid. Why do we know that it is haploid? Because assuming I did this correctly and used the same colors, which I did, this here refers to two sets of that specific chromosome or that specific DNA set. This is one set of it. Same thing here, two sets of it, one set. Two sets, one set. As we start to get into more complex, because it's half of the other one, does that matter? Yes, that's exactly it for the most part. That's exactly it. Okay, so hopefully that solidifies the understanding with regards to haploid and diploid. And then we can always come back to that to have a discussion on it if we still have some questions with regards to haploid and diploid. But haploid and diploid, a common theme you'll see come up many, many, many times throughout this lesson, as well as the next few lessons. So continue the good questions if you have them. Okay. There we go. So the next thing I want to talk to you a bit about is what's called karyotyping. And karyotyping allows us to analyze the number and characteristics of chromosomes in a cell. What happens if the diploid cell has two black strands, two orange, but one pink strand? Is it still considered diploid? It's diploid, but there's an issue with the genetic information, Jessica. It should have two sets of each. And you'll see in this karyotype what I mean by that, because we'll go over karyotyping in general. No, it wouldn't be a mutation. It would be what's called a non-disjunction. Mutations are where specific genes or DNA are altered. If it's missing a chromosome altogether, that's bad. Usually, that's bad. Or at least it doesn't lend itself to good things happening. Right? These are whole chromosomes, right? It's not just a set of genes or it's not just a piece of DNA. one whole chromosome missing is bad. Yeah, no worries. Good question. Okay, so karyotypes show chromosomes that have been stained and then arranged by size, the shape, and number. The pair of chromosomes are called homologous because they contain the same types of genes. So here we see 2 sets of 23 genes. Each set came from one biological parent's gamete. So here we got the green one is mom, and then this red highlight is dad. So green is mom, red is dad. Green is mom, red is dad. And so on and so forth, okay? Karyotype is the shape of the cell. No, karyotype is the process with which we isolate chromosomes. Don't mistake karyotype and prokaryote or eukaryote. Those are completely different things, Cool? Wait, isn't it 23 sets of 2? No, we have 2 sets of 23. One from mom, one set from mom. I'm highlighting the ones so dad red, mom green, so on and so forth. The highlighted greens are one set. One set of 23 from mom, one set of 23 from dad. Don't look at it as 23 sets of 2. Karyotype is a process. Yes, karyotype is the test or the process to isolate those chromosomes and arrange them. Wait, when we arrange it by size, shape, and number, what do you mean by number? By number is just like from largest, which is 1, to smallest, which is 23. So 1 being the largest ones, 2 being the next largest, and so on and so forth. That's what we mean by like a number. Does that make? Yeah, it's just size technically. Alrighty. So the other thing I want to spend some time talking about with regards to this, right? Green is one set from one parent, and I should say red, not gray. That's okay, I'll just do red. Bam. Green is one set from one parent, red set from parent 2. Okay, now the key thing here to realize is that chromosomes 1 to 22 are called autosomes. Autosomes. Chromosomes 23 are special. The chromosomes 23 are special. And they're special because they are called sex cells. They are singular or not homologous, meaning they can be different from each other. This is a key concept that I think a lot of students struggle with, and I'll reiterate it one more time. Where is it? Oh yeah, here. This point here, I will stress this a million times until the sun explodes for the entirety of my life. The paired chromosomes are called homologous because they contain the same type of genes. What does that mean? What does that mean? Who can tell me? You don't have to know it. If you don't know it, that's fine. Who can tell me what they think that means? Because it directly relates to this as well. But what does it mean by homologous chromosomes contain the same type of genes? What do you think it means? And if it's okay if you don't know. You're not supposed to know right now. Take a guess. Good guess, Matthew, but no, it's not from the same parent. They're from one from each set of parent, right? What I mean by that is the green and red one that I've highlighted at the front. No, only look at the ones in the up here. I'm only focusing on these ones here. Yeah. I'm only focusing on the ones I just highlighted. Amina, you're very close. Actually, no, you're technically right, Amina. Jessica, you're kind of like too. You're kind of right too. Olivia, you're on the right path, but go a little bit deeper. Also, I love that you're all messaging me individually and not spoiling it for other people. From both parents. Yes, that's a given. I will say that that is a given. So I'm looking at this highlighted orange point. The paired chromosomes, the ones that I've highlighted in orange here, one from mom, one from dad, they are what's called homologous because they contain the same type of genes. Why is that important? Or what does that mean, I should say? And if you're not sure, that's okay. I'm going to explain it from the entire unit and then some. But I just want to see what people think about it. You're right, Olivia. Well, kind of you're right. But why is that important? Like, what does that mean? And Amina, you were the closest one so far. So if you want to, I can give you credit for your answer. And then if you want, because you're missing a little bit of detail, but again, I'm not really expecting you all to know. So if everyone's kind of done guessing or thinking about it and wants me to put them out of their misery, so to speak. Okay, I'm going to talk about it now. So what does this mean? What does it mean they contain the same type of genes? So let me just get out of here for a second. Here we have one chromosome from mom, one chromosome from dad, or it doesn't really matter, right? This is chromosome number 1, let's say, or chromosome pair number 1. So in your set, your 23 sets, right? This is the first set. Your mom has, let's say, green eyes. Actually, let's go blue eyes. Your mom has blue eyes. This is the gene responsible for blue eyes, okay? Your mom also has blonde hair. This is the gene set responsible for blonde hair. Your dad has brown eyes. Where do you think that gene would be located on his chromosome that he gave to you? If your dad has brown hair, where do you think that gene is located on your dad's chromosome that he gave to you? If your mom has blonde hair here on this gene on that chromosome, no, we're doing the hair. We're doing the hair first. Your mom has blonde hair on, oh, what just happened? There we go. Good, Matthew got it. Let's see if anyone else can get it. Say, oh, good. Good. So if your mom's blonde hair is located on the bottom of that chromosome, because they are homologous pairs, this is the big idea, because they're homologous pairs, your dad's brown hair genes, and I'm oversimplifying it, but it doesn't matter. The brown hair genes are going to be right there. Now, this is important because you were born with brownish blonde hair, which makes sense because the genes located in the exact same spot in the exact same homologous pair can then be used by the cells to create the necessary genetic information or proteins or what have you that make you you. Okay, so we did the hair color. Now let's do eye color. Your mom's blue eyes are up there. Your dad also has blue eyes. Holy smokes. Where is that gene located? Where is that gene situation located? On your dad's chromosome. Good. And that is why homologous chromosomes is such an important concept, because the homologous pairs have the exact same genetic information in the exact same spot. Even though your mom might have blonde hair and your dad has brown hair, the genes responsible for that color are located in the same spot. How do you naturally select one gene over another, even though they are in the same place? Matthew, we get to that in lesson 5. Don't you worry. Why are there some people born with different colored eyes then? Don't worry, Daniela. That's the exact same question that Matthew asked. We get to that in lesson 5. or 6, sorry. No, not mutants. It has to do with heredity. And we'll talk about that eventually. But that depends on this stuff, right? In order to know why some people get brown hair or blonde hair or blue eyes or brown eyes, knowing this homologous pairing situation, that is dependent on our understanding later on, okay? So I need a check mark from every single one of you before I move on. Does this make sense when I mean homologous pairs? Give me a check mark in the participant section if you, for the most part, get what I'm talking about. Say that my mom has blue eyes and my dad has brown, and then son and the daughter both have brown eyes. What decides that? Yeah, we talk about that in lesson 6, Emma. If genetically it makes sense that both parents have blue eyes and someone is born with green eyes, how is that possible? We'll also talk about that in lesson 6, Daniela. Is it just chance? Kind of. Kind of. But that's why genetics is so cool, because we can talk about that stuff. But in order to understand that, we have to know what that means by homologous pairs, okay? We have to know the homologous pairs. Genetic history, kind of not really. Kind of not really. Because let's say, for example, your dad had green eyes instead here, right? Let's say, for example, your dad had green eyes here. Oop, hold on. What color were your eyes going to be then, right? And that's what we'll talk about in lesson 6. All right, so far we have 14 people who are good with this homologous pair situation. Hopefully it's more, and hopefully the other people are just too gun shy to give me a check mark. But this, I promise you I'm going to ask a question on this on the quiz. Like, without a shadow of a doubt. I will ask you to tell me about homologous pairs. I just created the question this morning, so if for no other reason than you like doing well on the quiz, please, hopefully you understand this concept. And if not, you can ask questions about it. All right. So karyotypes, homologous because they contain the same set of genes. However, now we're going to throw a little bit of a curveball. The 23rd pair here, they are not homologous, or at least not always homologous. Because in a biological male, the Y chromosome is technically smaller than the X chromosome. The Y chromosome is smaller than the X chromosome. So they are not homologous pairs. So in this example here, we have a biological male, an X and a Y chromosome. You can see that the X and Y chromosome are distinctly different sized, and therefore they are not homologous. So the sex chromosomes, or chromosome 23, are not homologous. Does that mean the chance of being a female is larger? No, not necessarily, Colin. 50-50. The Y chromosomes are smaller than the X chromosome for a biological pair. Correct. Yes. Sir, can I explain X? Yeah, so XX means biological female. If it was a biological female, this chromosome here, this is the X chromosome, this would be the Y chromosome. It would be homologous. But because it's a male, we have a significantly smaller Y chromosome. The genetic information for a biological male is smaller than it is for a female. What's the difference between autosome and homologous? Autosome are non-sex chromosomes. Homologous is the pairing of those chromosomes. So homologous was that example we looked at just before, where I talked about the brown hair and the blue eyes and how the DNA matches up on a homologous pair of chromosomes. Chromosome pair 1 have the same DNA, basically, just with variation. And then with regards to autosomes, autosome chromosomes are all homologous. So chromosomes 1 to 22 are all homologous. They are paired up, whereas chromosomes 23 are not. And this decides whether it's a boy or a girl. Correct, Faye? Genetically speaking, that is. And did that make sense with regards to homologous and autosome? That question? Or the difference between them? Alrighty. Let's take a look at how these gametes are produced, because this is the big meat and potatoes concepts that you're going to have to know for the quiz as well. In full detail, you will have to know meiosis. Okay, and it's a lot of information, and that's why I'm having you write and draw it down yourself. So if you don't have that sheet of paper handy yet, we're not quite there. That will kind of be your homework to copy it down in some concepts and ask questions about it. But you're going to need to write down and draw down the whole process of meiosis and understand it in its entirety. So without further ado, let's take a look at how those gametes are produced. Because remember, mitosis produces identical daughter cells to the parent. Okay, that 2n equals 46 is reproduced. That all of that genetic information is copied, and then a new set of that DNA is made for the daughter cells, and then it splits. Okay, and then it has 2 equal parts of chromosomal information that is the same as its parent cell. Now, if this is true, then how can gametes with half the amount of DNA be produced in an individual? Well, it's because we have to go through what's called meiosis. And meiosis is a 2-stage division of cells which produces gametes. It is similar to mitosis, but there are some subtle and big differences, really and truly. So the production of special sex cells is essential because they must contain half the amount of DNA that somatic or body cells contain. This ensures that the correct number of chromosomes for the zygote, because remember, those 2 gametes have to fuse their genetic information together to form that zygote. And that's the first cell of a new offspring. And then lastly, in humans, the gametes are egg and sperm cells. When egg and sperm cells meet, they form a zygote. So fill in this part of the note, ask questions. I'll give everyone just a couple of minutes just to get this down, because then we got to get into the big parts of this lesson, where we look at the outcome of meiosis and then the specifics of meiosis as well. Because then we can start to look at genetic variation, which is the whole reason why we study genetics. But in order to do that, we got to have a good, strong foundation of meiosis. Do octopus have eggs? Yes. Technically, yes. No worries. No worries. Okay. Any questions about that intro part? Because then we're going to start looking at the outcomes of meiosis in specifics. Alrighty, so let's take a look at this outcome of meiosis, because this isn't the specific details of meiosis yet, but it kind of paints a clearer picture as to how some of this process or these processes happen. All right, and we'll look at the more specifics later on. I've also added an explore learning activity for us to do for homework later today or over the weekend, whatever floats the boat down the moat, to kind of help us further review meiosis and the process. Okay, so there's two types of meiosis. First is spermatogenesis. This creates sperm, okay? We'll look at this one first, and then we'll look at oogenesis, which creates eggs. Now, spermatogenesis takes a somatic cell. So this here is somatic still, right? Because it's diploid, and it has a full set of its DNA, right? 46 total chromosomes. Now, it is not duplicated, okay? It is not duplicated in the classical sense, all right? Not duplicated in the classical sense. Because in order for that to happen, if it's duplicated, then we would just be in the same stage of what do you call it? Meiosis, or mitosis, if you will. And then that's not what we want, because what we want in that next stage of meiosis, we want it to have half the number. And then that genetic information can be duplicated, and then it can be reorganized and split up. So that way we have 4 cells with a single set of chromosomes only, okay? So this produces 4 sperm cells with non-identical genetic information. And this is the key thing here, right? It's gotta have non-identical information. Okay, questions? So again, it's not duplicated in the classical sense. It's not duplicated in the classical sense, okay? And I'll look at the specifics of that duplication later on, because you have to understand that it's slightly different from mitosis, but it uses the similar processes. Okay, whereas with oogenesis, same thing is happening, right? Same steps in meiosis is happening. However, we end up with what's called polar bodies. So it doesn't quite go through the same process as spermatogenesis as it does with oogenesis or egg production, because ultimately in spermatogenesis, it produces 4 sperm cells for every 1 cell that it's duplicated from. Whereas in oogenesis, it only produces 1 egg from that original cell, and the rest are what's called polar bodies that are recycled and then turned into eggs later on. Okay, take a breath. If you have a question, let me know. So far, we're okay, I think. Not too much detail thrown at us just yet. But this is a denser lesson, so, oops. Okay, I see a question. Let me just take a look. It's duplicated twice. Technically, yes. Technically, yes. And I'll talk about why that's the case later on, or what that actually means and how it works. So there are 4 sperms in total. 4 sperm in total. The 4 sperm. No worries, Stanley. Thanks for letting me know. Yeah, 4 sperm from that 1 egg that they all duplicated from. All right, let's take a look at how this genetic variation arises and why it's so crucial to why we're studying this. So introducing this concept of genetic variation during meiosis, it's important to consider it as a whole. Oh, 1 second. You can't see oogenesis? There, what about now? Can you see it now? Okay. Yeah, if you can always look after, it's a pretty straightforward step or stage in the context of relative to spermatogenesis, just because there's the duplication events don't really happen the same way. And it ends up with just one egg. But you can always come back to it later. Okay, genetic variation. What is genetic variation? Well, in meiosis, we have two things that can happen that allow for new genetic variation. During meiosis, new gene combinations are introduced, which creates that genetic variation. The first is called crossing over. Now, again, we talked about those homologous chromosomes, right? Once they are copied that first time, again, not in the classical sense because of this crossing over, once they are copied, they can switch sections. So tetrads form by the pairing of two homologous chromosomes that have been copied. Crossing over is the exchange of information between those homologous chromosomes. Crossing over happens at multiple locations across the chromosome. There are many what's called chiasmata or crossing over locations. Remember when we talked about that homologous chromosome pair at the beginning? This is why it's so important that those gene sections that are responsible for the exact same thing are located in the exact same spot. Because eventually, in meiosis, they can cross that information over and switch parts of their DNA with each other. So some DNA that was your mom's goes to your dad's chromosome and vice versa. And this, my young students, this is where the big ideas of genetic variation start to come into play here. Because not only can you have that rearrangement in meiosis of which cells get which pairs, but now you have the homologous chromosomes sharing genetic information with each other. So take a second to write this down to digest it. It's a really big concept. I'm really hitting you hard with the big ideas here today, but that's okay because this is the last set before we go into meiosis as a whole review. So it's okay if you're not 100% sure. I also have a meiosis review. Actually, I should label that properly. 1 second. What lesson are we on? Lesson 2. So take a second and write this down, and then we'll talk in more detail about meiosis as a whole. Okay, questions about crossing over yet? Or about the idea of crossing over? As I see many check marks appear saying that you're good. Thank you for letting me know, folks. Or were those old check marks? They might have been old check marks, but they hopefully are new check marks as well, interspersed in there. All righty. I'll move on to the next section. The next section connects the idea of still, again, genetic variation, but instead of the crossing over, we look at something called random assortment. So let me just get my, now, of course, it's decided to die on me. Okay, got to reshare my screen. 1 second, folks. There we go. All right, so we have crossing over as one way with which genetic variation is introduced. And then we also have random/independent assortment. These two words are interchangeable. Random and independent assortment are interchangeable. Ooh, wrong color. So random and independent assortment, interchangeable. So this is when homologous chromosomes align during the processes of metaphase 1, as well as separation during anaphase 1. They will assort randomly, meaning each daughter cell that can receive either chromosome from that pair, red or blue. So when you take a look at just this diagram here, right? Oop, that's not what I wanted. When you take a look at this diagram here, okay, we have all three of the reds organized up top and all three of the blues organized on the bottom. However, in the diagram to its right and every other diagram, we have some variation of random assortment, meaning some of the DNA from mom is with dad, some of the DNA from dad is with mom, and vice versa in randomly assorted organization. So each thing that I'm highlighting shows you that the cell that is created as a result of this process of meiosis 1 will have different genetic information due to this random assortment. The number of possible combinations can be calculated by 2 to the n. So since humans have n equals 23, the number of possible combinations that can occur for sperm and egg gametes is 2 to the 23, or 8,388,608. That's how many different combinations of sperm and egg can occur just by random and independent assortment. This does not account for crossing over or random fertilization of gametes as well. So no two individuals ever in the history of humanity will ever receive the same information because DNA is combined differently each and every single time. So take a second here to digest this. This is the big reason why genetic variation is so cool, because no two humans ever will ever be the same, simply due to the fact that random assortment combined with crossing over, combined with random fertilization as a whole, it yields the possibility of hundreds of billions of trillions of different combinations of human beings, more so than ever will exist, have existed, or potentially will ever exist. So take a second here, ask questions if you're not sure about what this random assortment means. And I'm more than happy to talk about the diagram as a whole if you need more elaboration, because then we can start to talk about meiosis as a process. But is it theoretically possible after millions of years? No. Hundreds of billions of trillions of humans need to be born. So unless we populate every single star in the galaxy and live for millions of years, that number will never be realized. And even then, the number is dependent on the number of humans, right? As the human population increases, so too do the number of possible outcomes. They're proportional to each other. So if the population stayed the same and the DNA variation didn't change, then yes, it's possible. But because that variation continues to change as a result of more humans being born, it just keeps getting bigger and bigger. So no, I would say no, it's never going to happen unless with identical twins. Or the theoretical possibility is like less than 0.0000001%, I should say. Because technically anything is theoretically possible, but whether it happens or not. All right, questions for random or independent assortment or for crossing over? Two big ideas for this lesson. I see no questions. I see people leaving. Oh, bother. Okay, so your homework now is I would like for you to copy this diagram down. I will go over it in more detail on what day? Tuesday? No, I'll go over it in more detail now. We got time. We got time. We'll go a little bit longer this week, but that's okay. Or today. That's okay. We got at least we can get through the first little bit of meiosis. So I will wipe this all out because it's a lot of information and we'll go step by step. Okay. All righty. So let's take a look at the step-by-step processes of meiosis. And if we don't get this done, we can always continue next week. You can always start. This is where at the beginning of class, I said you need a sheet of paper. This is the time where you need that sheet of paper so you can write this down or draw the diagrams or what have you. Okay. So for this example, I am using the chromosomal data that will be 2n is equal to 4. Just to continue to keep it simplified, 2n equals 4 in this organism. So non-human organism. Now, the processes of meiosis are very similar to mitosis, okay? In this example, we have 2 sets of chromosomes, red and blue, and they are chromatin, long and thin, until they are duplicated in that S phase. And that duplicated chromosome, or that X-shaped chromosome, we count the number of centromeres to count the number of chromosomes. And this is why it's important for us to realize that that replication process is different from mitosis, because we're counting the number of centromeres here to determine the number of chromosomes. So when you look at the number of centromeres, here we have one, let me get a different color actually, pink. We have one, 2, 3, 4. Can everyone see that? It's kind of small, but my hope is that you can at least see some of it. Or maybe I'll add a little highlighter. 1, 2, 3, 4. So that's how many chromosomes we have. Most people get confused. They're like, well, it duplicated, so we have technically 8 chromosomes. No, we're counting the number of centromeres here to ensure that stability with regards to our understanding of the number of whole actual like sets of DNA, not just the set plus the copy, just the set itself. Does everyone understand what I mean by that? I want to spend some time just talking to make sure that you understand what I mean by that, because that can be a little bit of a weird concept to understand. Have you all checked out already? Yeah, kind of. All right, let's stop here then. Let's stop here. We'll go over this process on Monday then. I think it's more important that we stop here. If I'm just talking to a blank slate or a blank wall, it's pretty useless because this is super important. So we'll stop here. We only have about 10 minutes left of class, so you can work asynchronously. If you would like to fill this note in over the weekend, I strongly recommend that you do that. But I will go over it on Monday because really and truly, it seems that you've all lost a little bit of steam on a Friday. And I think it's important that when I speak to you about this, that you're at least with me, even a little bit. And it feels like, even though I can't tell 100%, it feels like you're not. So we'll stop here. I'll give you all a bit of a breather. Over the weekend, still take a look at that homework I posted in your outline though, okay? Because it will be very important come Monday, because I'm going to assume you looked at this over the weekend. Okay, I'm going to assume you looked at these stages over the weekend. So when I cover it, I'll just focus on the big important ideas. Okay, so that's your homework. I'll put it even in the outline. Where's my outline? You got a busy weekend. Please look over the meiosis chart. Okay, please. Oh, also one final thing before you go. Can you please quickly, it's 2 questions. Fill in this Google form for me before you all head out for the day. It's just a quick little check-in for lessons 1 to 3. I'm asking about what assessments you have next week, and I'm asking how much you've studied so far this week. 2 whole questions. Whether you do that right this instant or over the next 5 minutes when you check other schedules, that's perfectly fine. Please take a look at this Google form. It is very crucial for your assessment next week. Please give me honest feedback. because it will help me schedule your quiz for next week to a correct date and time. So please do that. Otherwise, if you're planning on working asynchronously for the remainder of class, by all means, head on out. If you want to stick around and ask questions about meiosis, by all means, hang out, ask some questions, do that Google form. And heck, folks, I know it was a lot of information today. So please try to digest it, look at it over the weekend, and I'll see you all on Monday. All right, folks? You're welcome, folks. Have a great weekend. Enjoy, everyone. Take care. We'll see you on Monday, folks. Don't forget to do that Google form. Take a look at that meiosis processes, please. Take care, everyone. Bye.
Have a nice weekend.
You too, Rocco. I think that was Rocco. Bye,
Bye,
folks.
Mr. Kudasal. Have a great weekend.
Bye, Matthew. You as well. Take care. Have a good one, Daniela. Bye, Eva. Bye, Olivia, Mateo, Jessica, Katie, Sarah. Bye, Carson. Sir, how can gametes with half the amount of DNA be produced by an individual? Yeah, we're going to talk about those steps on Monday, Fayang. You can also take a look at the Explore Learning activity that I posted to our Explore Learning class on explorelearning.com. But that's this process of meiosis that I didn't go over today because we kind of don't really have time to go in full detail. But I'll spend more time talking about it Monday. You can also look at it over the weekend. Okay, but that will be for Monday. People ran out of steam today. There's a lot of information for everyone. Only one egg at the end of oogenesis, but on the notes there are two, one big, one small. Yeah, the smaller one gets is called a polar body, and that gets reused to become an egg later on. So I'll show you 1 second. Let me go back to that. You're talking about this note here, right? Yeah, so this is the egg. And then this, oops. And these are what's called polar bodies. And these polar bodies get recycled and reused in this process, and it's a cycle and it repeats. No problem, Faye. Chromosomes 1 and 2 are called autosomes. Chromosome 23 are called sex chromosomes. They are the sex chromosomes, for lack of a better word. No problem. Yeah, there's sex chromosomes. I'll put that here. All right, folks, those of you still kicking around, it is 10:20. I'm going to close up the Zoom. I hope you have an excellent rest of your day and weekend. If you have any questions, we'll talk about it more on Monday. Take care, everybody. Enjoy your weekend.